here in relation to the amount in the wild type controls. But the signal for the NR2B mRNA was not upregulated in the brainstem and thalamus or in the cerebellum. So their targeting seems to have got them what they wanted. By expressing the GluN2B subunit under control of the CAM kinase 2 promoter, they're getting elevated expression that seems to be confined mostly to structures of the forebrain neurons. I also want to drive home this message that if you were to create a new kind of transgenic animal, it's really, really, really important that you do a rigorous characterization of that animal like this group did, like Tang and Xian did. I want you to understand that it's really important to examine these animals very carefully. And I'm going to try to illustrate that with the image on this slide. So in another experiment, and I honestly don't remember where I got this particular photograph, I've had it for years, a kinase called FIN was transgenically overexpressed in mice. And, well, those mice were cognitively impaired. And when the investigators examined the brains of the mice, they found that the granule cells of the dentate gyrus and the CA3 pyramidal cells, all of these marked by arrowheads, were overabundant. You see the way the dentate gyrus should be a nice smooth structure here, and the same with the CA3 pyramidal cells. They should line up very nicely, and we have all of these additional cells in the dentate gyrus and the CA3 region here. So, as with most or, well, perhaps all proteins, maybe especially kinases like FIN, well, FIN serves multiple functions in cells. In this case, it turns out that FIN is actually involved in apoptotic mechanisms during early development. So the impairments in these mice could be due to a developmental error of apoptosis, rather than any specific difference in function of overexpressed FIN in the adult mice. So Tang and Xian did due diligence and found that their Doogie mice appeared normal in so many respects, but were actually overexpressing GluN1, GluN2B subunits in adulthood in forebrain regions. Okay, so let's look at the actual function of these overexpressed GluN2B containing NMDA receptors in the Doogie mice. Electrophysiological examination of the Doogie mice revealed that these transgenics actually didn't differ from wild-type mice in the juvenile stage at postnatal days 10 to 14. And this makes good sense because the juvenile mice were also expressing high levels of GluN1, GluN2B complexes at that age anyway. Okay, so it's well known that NMDA receptors have long opening times. I showed you that on a previous slide, with a lot of calcium coming in and those ion channels staying open for a prolonged period of time compared to those that have the GluN2A subunit. But at postnatal day 10 and postnatal day 14, we don't see any difference in the decay times between the wild-type mice and the Doogie mice at postnatal day 10, or the same thing at postnatal day 14. However, when comparing the decay times between the adult Doogie mice and the adult wild types at day 18, now we see some differences. The Doogie mice continued to have a high peak in terms of the calcium influx and prolonged opening in comparison with the receptors in the adult wild-type mice. So the calcium influx in the Doogie mice turns out that it was actually about 4 times higher in these Doogie mice than it was in the wild-type mice. The decay time in the Doogie mice is longer in the adult stage than it is in the wild-type mice, and the peak amplitude in the adult Doogie mice is higher than it is in the wild-type adult mice. So clearly it seems that adult Doogie hippocampal cells have more GluN2B subunits per synapse than adult wild-type mice have. Now, additional electrophysiological tests showed that measures of presynaptic function were normal in the Doogie mice. In this test, a depolarizing pulse is administered to the Schaefer collaterals, and the CA1 pyramidal cell, EPSP, is recorded. And then at intervals ranging from about 10 to 400 milliseconds, or 10 to 400 thousandths of a second pulse is delivered, and a second EPSP is recorded. The first pulse causes calcium influx, and the synergistic effect of the residual calcium added to the second calcium influx from the second pulse produces a facilitation of the response to the second pulse. If there are presynaptic enhancements like increasing voltage-gated calcium channel function or decreasing voltage-gated potassium channel function, then the facilitation will be greater. This is called pre-pulse facilitation, or I'm sorry, paired pulse facilitation. Paired pulse facilitation. And this wasn't seen in the Doogie mice. They had normal paired pulse facilitation looking just the same as in the wild-type mice. So the measure of presynaptic function looks normal in these mice. All the enhancements appear to be happening postsynaptically. It was also noted that NMDA receptor-mediated EPSP responses to single pulses were greater in the Doogie mice than the wild types. And this again was due to the NMDA receptor expression, as the AMPA currents were normal. So all of this simply adds up to the observation that the adult Doogie mice had a super responsive set of NMDA receptors in comparison with the wild type mice, with no accompanying changes in AMPA receptors or other measures of synaptic function. So let's get down to the nitty-gritty of it and look at a measure of plasticity. As you might have guessed by now, when Schaefer collateral to CA1 tetanic stimulation was applied, the Doogie mice exhibited greater LTP. In this case, a mild form of tetanic stimulation was applied so differences could be readily observed. If they had used very strong, high-frequency stimulation with lots of trains of stimulation, of course, you would have maxed out potentiation in both sets of animals, and it would have been harder to see the differences. But in the graph on the left, you should be able to see that the adult hippocampal sections from 2 lines of Doogie transgenic mice developed strong and enduring LTP when given just 1 train of 100 Hz stimulation. And in contrast, the wild-type mice only developed a transient expression of LTP that you would expect under these circumstances. And if we turn to the graph over on the right, we can see that even 10 Hz of stimulation given for 90 seconds that did nothing to plasticity in the wild-type sections actually produced LTP in both lines of Doogie transgenics. So clearly both lines of the GluN2B overexpressing Doogie mice had enhanced potentiation when Schaefer collateral inputs to CA1 pyramidal cells were stimulated in sections from the adult mice. But on the other hand, long-term depression, LTD, didn't differ between the Doogies and the wild types. The graph on the left over here shows that 1 Hertz stimulation administered for 15 minutes, kind of a standard LTD inducing protocol, produced the similar LTD between the wild types and the transgenics. So the graph that's over on the right actually summarizes a whole range of different stimulation parameters and shows that the Doogie mice with enhanced conductivity through their GluN1, GluN2B receptors showed enhanced potentiating sensitivity to the CA1 to the Schaefer collateral, the CA1 pyramidal cell synapses from, well, about 10 Hertz up to 100 Hertz stimulation. But actually normal responses for any kind of stimulation at lower frequencies than that. And I can just about hear everybody fidgeting in their seats thinking about the next question. I can just about see that student in the back of the class with his hand up going, oh, 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 and asking the question, what about behavior? So yeah, let's do that. As you might expect, because otherwise it really wouldn't be a very good story, would it? While the Doogie mice exhibited superior performance at the novel object recognition test. The graph at the upper right shows that they didn't differ from the wild types in terms of their exploration of the stimuli at baseline. The graph at the lower right shows that when the mice were tested with a novel and a familiar object 1 hour after the training session, they did equally well in selecting the novel object and exploring it, performing just basically the same as the wild type mice. But when tested 24 hours later, the Doogie mice appeared to have better recall of the familiar object as they explored the novel object much more than the wild type mice did. And the same was true when tested after 3 days. But, well, all the mice failed and discovered and explored both the novel and familiar objects at chance levels equivalently if they were tested 1 week later. So clearly in terms of this test, the Doogie mice did a lot better than the wild type mice. But if you ask my honest opinion, based on the outcomes of this experiment, I don't think they're quite ready to take their medical boards just yet. But, well, they also do better than the wild type mice in contextual fear conditioning. The top graph shows that the wild types and 2 transgenic lines exhibit about equivalent freezing when exposed to the shocks in the training session, but the Doogie mice did better in recognizing the context and freezing in it when tested 1 hour later, or, well, 1 day later, or even 10 days later. Genius. And similarly, the Doogie mice did better in cued fear conditioning when that was assessed 1 hour, or 1 day, or 10 days after the training session. So here's a little puzzle for you. If you've taken an intro psychology course, which I know most of you have, you learned about extinction. Now, in case you haven't taken that course, I'll quickly explain. If you are a mouse and you learn an association like a contextual or a cued fear conditioning, and then you're presented with multiple trials where the association is not supported, like you're placed back into the context but there's no foot shock, or the tone that served as a cue for foot shock is presented over and over, but no foot shock is delivered, your freezing response will diminish across trials. This is known as extinction of the response. So, without looking at the graphs, who do you think should extinguish faster? Remember, the Doogie mice learned the contextual and cued associations more strongly. So who's going to extinguish more quickly? Well, the Doogie mice, of course. And yes, it does make sense. Even though they learned the association more robustly, they extinguish more quickly, because extinction is not forgetting the association. It's new learning. It's learning that the context does not predict foot shock. And it's learning that the cue does not predict the foot shock. In fact, we now know that extinction is impaired by administration of NMDA receptor antagonists like APV, or disrupted by administration of actinomycin or anisomycin. So the Doogie mice not only learned the initial associations more quickly and more strongly, they also learned the new circumstances in which the context and cue no longer predict the foot shock more quickly. Well, maybe I should get me a mouse doctor after all. And finally, we have the Morris Water Maze. So Tang and collaborators performed standard spatial learning tests with the Morris Water Maze and found that the Doogie mice learned more quickly across the training sessions, although the wild-type mice basically caught up to them by about the 5th or 6th session. So the faster learning is also demonstrated by the middle graph that shows that the Doogie mice spend more time in the target quadrant than the wild-type mice do in a probe trial after only 3 training sessions. But then, after the 6th training session, the Doogie and wild-type mice both exhibit a robust preference for the target quadrant. So that sums up the story with the Doogie mice. By overexpressing the GluN2B subunit of the NMDA receptor, these mice had greater potentiation in LTP, and they appeared to learn a variety of tasks more quickly, more readily than the wild-type mice did. So here's another puzzle for you. If this actually explains why, or at least in part explains why, juvenile animals can learn so much more readily in a variety of contexts than us adults can, and why do we ever give up these GluN2B subunits, and why do we replace them with GluN2A? Well, it looks like evolutionarily there's a good reason for that too. Because calcium coming into cells in too large amounts can be toxic. And so across aging, it looks like we really wouldn't want to have that enhanced calcium influx going on and on and on. It may be suitable for the young juvenile stage, but you need to be able to survive with an intact brain to the reproductive age in order to pass on your genes. So perhaps you wouldn't be doing that if you continued to have large amounts of calcium influxing into cells every time there was a salient event. So what's good in the juvenile stage actually may not be so well suited to adulthood. So I've been talking a lot about NMDA receptors and the famous Doogie mice. But I promised at the beginning that I'd also show you something about genetic manipulations of AMPA receptors and what these manipulations have taught us about the circuits that underlie learning and memory. So here goes. This experiment is based upon a very simple knockout of the gene that encodes the GluA1 subunit of the AMPA receptor. This is not a tissue-specific or inducible kind of manipulation. They just grossly knocked out the GluA1 subunit of the AMPA receptor overall. And so you can see in this left hemisection of the rat brain, of the mouse brain over here, representing a wild type mouse, that there's abundant expression of the GluA1 subunit in stratum radiatum and stratum oriens of the hippocampus, as well as lots in other areas of hippocampus, and maybe some over in amygdala over here, and in some subcortical regions and other regions of the cortex. The GluA1 subunit in the wild type mouse is abundantly expressed in many different structures. But if you look at the other hemisection here, representing the GluA1 knockout, you can see that the receptor is completely missing. There's some counter stain on the section so you can see the structures, but there is no expression of the GluA1 subunit whatsoever. And, well, it really doesn't take a genius to realize that if you wipe out the GluA1 subunit of the AMPA receptor, then Schaefer collateral to CA1-LTP should be profoundly impaired in these mice. And so actually it is. Following brief strong tetanic stimulation, the wild type mice show a robust and enduring nice LTP, whereas the GluA1 knockouts show no potentiation whatsoever. But I want to show you a cool kind of other experiment in the GluA1 knockout mice. So first, I'm going to remind you that the GluA1 homotetramers, shown here, and again, VoiceThread messes up my little diagram a little bit, but it's not too bad. These are the GluA1 homotetramers in comparison with the GluA1-2s and GluA2-3s that generally populate the synapses. So I want to remind you that the GluA1 homotetramers are rapidly and transiently inserted into the postsynaptic membrane after tetanic stimulation. So the fact that they're missing could account for the lack of LTP in this study. But there's a bigger implication on the next slide. For this one, I need you to think all the way back to the first chapter. I need you to remember some of the information I gave you about the workings of the radial arm maze. Okay, so I'll quickly remind you. In the radial arm maze, you can bait the arms of the maze with food. So it looks like in this depiction of the maze here, we've got arms 1, 4, 6, and 7 baited with food. A mouse placed onto the maze has to select which arms it will visit. And once it has visited an arm, it shouldn't return there, as that arm would be empty. The animal has already retrieved the food. Now, during training sessions, the same set of arms can be baited over and over again, and the other arms are not. Then in the test session, the animal can visit the arms freely to obtain food rewards. Two types of memory are assessed in the test session. One is reference memory, and the other is working memory. Reference memory is demonstrated when the rat only visits the arms of the maze that contain the food pellets, which they actually learned in the training sessions. Entry into an arm that was unbaited in the training sessions would be a reference error. It's as if they do not have a recollection of which arms were baited in those previous sessions. Working memory is demonstrated when the rat enters each baited arm a single time during the test. Because re-entry into an arm from which the food was already retrieved, that would be a working memory error. And so it turns out, as you can see on the graph, that GLUE1 knockout mice are impaired in reference memory. They make more errors in this long-term memory. And this long-term memory perhaps failed because of the overall failure in synaptic plasticity, as was demonstrated in the LTP experiment on the previous slide. But perhaps more interesting is that the GLUE1 knockout mice also failed in terms of working memory, and miserably so. You can see that the wild type mice here are extremely efficient and virtually never re-enter a baited arm once they've retrieved the food. And it makes sense they're so good at this because you only have to keep the spatial pattern in your brain for seconds or at most a few minutes. So knocking out GLUE1 subunits disrupts working memory. In other words, working memory in these mice requires the rapid insertion of GLUE1 containing AMPA receptors into the membrane. So this is an animal model of working memory, something akin to me telling you to remember my phone number is 8675309. And no, that's not my actual phone number. It's a phone number from an old song by Tommy Tutone. But I digress. If you try to remember a phone number so you can bang it into your phone, or you try to do a little mental arithmetic to figure out which brand gives you more ounces of yumminess per dollar at Publix, these are working memory tasks. And the inference of the radial arm maze result is that working memory requires rapid insertion of those GLUE1 homotetramers that we saw in LTP induction somewhere in the brain, perhaps in dorsolateral prefrontal cortex. Wouldn't it be fun to go looking for where those receptors are actually being inserted in a working memory task, like the radial arm maze? Again, anybody want to do a dissertation project?